Hi guys, I am Sarang Nantra Pawar. Today in this video we are looking the information about the DNA and DNA structure. Basically, what is the DNA? DNA is the deoxyribonucleic acid. The full form of the DNA is deoxyribonucleic acid, and the structure of the DNA and the name. When we correlate the structure of the DNA and name of the, that is the full form of the DNA. It is easy to understand the whole structure of the DNA. The first of all, from the DNA is made up of the nucleotides. It is the polymeric chain of the nucleotides. Then what is the nucleotides? Nucleotides is the structure accompanied of the phosphate groups, that is the PO4 minus. After that, the 5 sugar, that is the 5 carbon sugar, that is the pentose sugar. It is basically pentose sugar is of two types. The first type is the pentose sugar. In the DNA there is the sugar is present in 2-deoxyribose sugar, and in the RNA there is ribose sugar, the ribose sugar. After that, the third part, that is include, that is contained in the nucleotide, that is the nitrogen base. There are two types of nitrogen base. The one is the purines and another is the pyridines. Purines are the double ring structures, that contains adenine and guanine. And pyrimidines are the single ring structure that contain cytosine, thymine or uracil. These are the, and in the basically in the DNA there is no uracil is present, only the cytosine and thymine is present in the that is the pyrimidines. After that, one question is rises, that is the what is the nucleosides. Nucleosides are the groups, it does not have the phosphate group, only the 5 carbon sugars, and the nitrogen bases contains the nucleosides. Nucleoside plus phosphate group is equal to nucleoside plus phosphate group. So we can call the nucleoside plus phosphate group is equal to nucleotides. Bonds, after that we are looking about the bonds between the bonds of DNA. The glycosidic bond is between the carbon, that is the the bond between the nitrogen bases and pentose sugar. That is the helical structure that around the helical structures external part that is detected, that is the pentose sugar, and that is the bond between them is the glycosidic bond. After that, the phosphodiester bond, it is the bond between the adjacent nucleotides connected together from a long polypeptide chain, that is the long polymeric chain. The bond between adjacent nucleotides connects together. The two or more nucleotides are connected by the phosphodiester bond. And after that, the hydrogen bond is the bond between the two nitrogen bases, that is the A or T, G or C. These are the bonds between the nitrogen bases, that is the hydrogen bonds. After that, we are looking about the, after that, in the DNA, the adenosine is connected with the thymine by double hydrogen bond, and the guanine is connected with the cytosine by the triple bonding structure, or detected by the triple bonding, that is the guanine is connected to cytosine, and adenosine is connected to thymine. This is detected by the double bonding. This is the bond between the DNA, and the main function of the DNA is the expression of the genomics, and the production of the proteins and the various functions of the DNA, that the maintaining the body temperature, म्हणजे irregularly DNA maintains the body function and controls the whole body system. This is the main function of the DNA. Thank you so much. We will meet soon.